Good morning, students. Today's session is on nucleotide metabolism. So, the competencies addressed are BI 6.2, BI 6.3, and BI 6.4. The specific learning objectives for today's sessions are: at the end of this session, you will be able to explain the digestion of nucleic acids in the body and their significance, enumerate the sources of carbon and nitrogens of purine, explain the regulation of purine nucleotide biosynthesis, enumerate the inhibitors of purine nucleotide biosynthesis, describe the purine salvage pathways and their importance, and finally, you will be able to discuss the causes, manifestations, and biochemical basis of these manifestations in Lesch-Nyhan syndrome. Now we will have a quick recap of nucleotide chemistry. Nucleotides are nitrogenous compounds consisting of a nitrogenous base, a sugar, and a phosphate. If a compound contains only nitrogenous base and sugar, they are termed as nucleosides. So the difference between nucleotides and nucleoside is the phosphate. Nucleotides will have a phosphate group in them. Now coming to the nitrogenous bases, there are two classes of nitrogenous bases, purines and pyrimidines. Purines, they have 9-membered heterocyclic nitrogenous ring in them. Example, we have adenine and guanine. Coming to pyrimidines, they are 6-membered heterocyclic nitrogenous rings, and examples are uracil, thymine, and cytosine. Coming to the sugar, so ribose sugar is present in RNA and deoxyribose is present in DNA. So with this background, let us learn about purine metabolism. Purines do not contribute to energy synthesis as that we see with proximate principles. The proximate principles are your carbohydrates, proteins, and fats. So purines, they don't release energy. Apart from this, these nucleotides are not incorporated into RNA or DNA. So unless they are given intravenously. So this shows that purines and pyrimidine nucleotides, they are dietarily non-essential. That means whatever dietary nucleotides we consume, they are normally metabolized to the individual components like bases, sugars, and phosphate in the gastrointestinal tract. So the purine bases in the intestinal mucosal cells after absorption are oxidized to uric acid and they are excreted in urine. So coming to the degradation of nucleoproteins, the nucleoproteins are acted upon by the HCl and the pepsin in the stomach to release the nucleic acids from proteins. Now these nucleic acids are acted upon by pancreatic enzyme, that is nucleases. We have DNAs, that is deoxyribonucleases for DNA, and we have RNAs, that is ribonucleases for RNAs. So they act on nucleic acids to release nucleotides. Now these nucleotides are acted upon by nucleotidases. These are intestinal mucosal cell enzymes. They release the phosphates and the nucleotides are converted into nucleosides. Now these nucleosides are acted upon by nucleosidases to convert it into base and ribose. Now these base, the purines or pyrimidines, they are absorbed into the mucosal cells and they are degraded into their respective catabolic products and they are excreted in the urine. In the previous slide, we saw that dietary nucleotides are non-essential and they are degraded into corresponding catabolic products. So that means that body has to synthesize purine nucleotides. So biosynthesis of purine nucleotides can happen through de novo synthesis, that is synthesis of a new purine nucleotides, or it can happen through salvage pathway where they use or utilize the existing nucleosides or purine bases. So there are three processes that contribute to purine nucleotide biosynthesis. If you see in the order of decreasing importance, the first one is synthesis from amphibolic intermediates via de novo synthesis, followed by phosphoribosylation of purine bases through salvage pathway, and further followed by phosphorylation of purine nucleosides, that is again a part of salvage pathway. So these are the three processes by which body synthesizes purine nucleotides. Now we will learn about de novo synthesis of purine nucleotides under these headings. So first we will see what are the sources of carbon and nitrogen of purine ring. Then we will just outline the reactions of de novo synthesis of purine nucleotides, followed by how this pathway is regulated and what are the inhibitors or clinical applications of the drugs which inhibit this purine biosynthesis. Now we will see what are the sources of carbon and nitrogens during the formation of purine ring. Already in chemistry of nucleotides, you have learned that purine ring is an aromatic heterocyclic ring and it is a 9-membered compound, and the numbering of this purine ring is anti-clockwise direction. So if you see the numbering, you can see it is 1, 2, 3, 4, 5 and 6. So it is in anti-clockwise direction. So many compounds contribute to the formation of this purine ring. So we will see now what are the sources of these C and, that is carbon and nitrogen. So the N1 is derived from amino group of aspartate. C2 is derived from N10 formyl tetrahydrofolate. Then N9 and N3, they are obtained from amide nitrogen of glutamine. So it is glutamine. Coming to carbon 4, carbon 5 and nitrogen at 7th position, they are contributed by very important amino acid, a simple amino acid, that is glycine. So the C4, C5 and N7, they are derived from glycine. The C6 is from carbon dioxide and C8 is from N5, N10 methanol tetrahydrofolate. Few books give the source of this C8 as N10 formyl tetrahydrofolate also. But the standard books, they provide the information as N5, N10 methanol tetrahydrofolate. So now you can see the formation of this purine ring requires aspartate, glycine and glutamine along with very important vitamin, that is folic acid. So before going to the biosynthesis of purine nucleotide, we will just see what are the important points to be remembered in biosynthesis or de novo synthesis of purine nucleotide. So the first intermediate formed in this biosynthesis is PRPP. So PRPP stands for 5-phosphoribosyl 1-pyrophosphate. So the synthesis of PRPP is not considered as a step in biosynthesis, but it is a preliminary step or a preparatory step for the formation of purine nucleotide. So the purine ring is later on assembled on the ribose 5-phosphate. So whatever 9-membered ring is there, actually it is step by step, it is built on ribose 5-phosphate. The first purine nucleotides to be synthesized is inosine monophosphate. Liver is the major tissue site and it is in the cytosol where this de novo synthesis happens. But there are some tissues and cells like brain, RBCs and polymorphonuclear cells, they cannot produce purine because they lack glutamine PRPP amidotransferase. So now let us see the preparatory step of purine nucleotide biosynthesis. The preparatory step is nothing but the synthesis of PRPP molecule. So if you see the reaction, this is actually an activation of ribose 5-phosphate molecule. Ribose 5-phosphate is an intermediate of HMP shunt. So the activation of this ribose 5-phosphate to 5-phosphoribosyl 1-pyrophosphate is catalyzed by PRPP synthetase. And for this reaction, ATP is a requirement. So after releasing the pyrophosphate from ATP, ATP is converted into AMP. Apart from this, PRPP is also utilized for pyrimidine nucleotides through salvage pathway and also required in niacin nucleotide synthesis. So once we have PRPP, now the purine ring is assembled on this PRPP. So that's why they say that this is not actually a step of de novo synthesis, but actually a preparatory step where there is activation of ribose 5-phosphate to PRPP.